and like we would go to softball like tournaments, we would always eat Nutella. And I ate so much Nutella that I would get like sick. Like I wouldn't eat it any, like, not that I would get sick because I ate so much of it from playing. It's like I couldn't eat it anymore for like
you
years.
got sick of it.
Yeah, for like years I couldn't stand it. Everybody was like, Nutella, Nutella. And I was like, ew, get that away from me.
Yeah.
It's like my phobia to, what is it? The silly string. Oh, I hate that stuff.
That was so random.
Ooh, we could do a phobia of ours for a bonus question. We could figure out what our phobia is and you can learn more.
Maybe that'll be for exam 4. It's a good idea. Or what weird snack do you eat? Or what food can you no longer eat?
Yeah, an extra bonus if we can make you laugh, because I can make you laugh with a phobia.
Oh my God.
I got phobia stories.
All right, well, yeah, this is a great photo. That's a healthy baby right there. That's what you want. My kids have never been that big, but it's so funny because a good friend of mine, her and I have been pregnant twice at the same time. And so our daughters are 5 weeks apart and our sons are 3 weeks apart. And her babies like are probably half the weight of my babies. I don't know why. We make big babies and they're heavy and they're chunky. And her baby's always so teeny. And so we laugh about it. So I send her this picture. I say, this is my baby or your baby? But anyways, that's a happy Thanksgiving, baby. Okay, lipid metabolism. So I'm going to keep bringing you back to this figure and this chart because it just kind of helps orient. And really, one of the main things I want you to take away from this course, of course, you're not going to remember every intermediate and every reaction in glycolysis. You're not going to remember everything we talked about in beta oxidation. That's not the details. You'll probably know it for exam 4, but for the rest of your life. But I want you to walk away with an appreciation for how everything is connected biochemically and how things feed into each other. So if you could walk away with this overall grand scheme of how digestion breaks down the macronutrients of our food, what that feeds into, what molecules are really important, pyruvate, acetyl-CoA, and how this feeds the citric acid cycle, electron transport, like that is a win for me. So we have already covered down here. We know we've covered, this was all chapter 21, citric acid cycle, electron transport, oxidative phosphorylation. Yesterday, we covered glycolysis and how glucose is metabolized to glucose 6-phosphate, and then how versatile that compound is. Glucose 6-phosphate, it can go several ways, okay? It can make glycogen. We could use it to make ribonucleic acids or NADPH. We could use it to later on synthesize lipids. We can run it through glycolysis, okay? So there's several things there. And then now today we're looking at lipids. So what happens when we have lipids? We know we've talked about lipids, chapter 23. There's like 7 different classes of lipids. You have your waxes, your triacylglycerols, your sphingomyelins, eicosanoids, steroids. We looked at all those different classes, okay? And we talked about the biological significance of those. Now what happens when those are digested and how do we absorb energy? We keep saying fats are our main source of energy. Fats are a huge source of energy. Now today we're gonna look at how we derive energy from the metabolism of fats, okay? So there's gonna be a few different parts. Number 1, we're gonna look at the digestion of fats. Because what do y'all know about fats? Fats, or I should say lipids, they're not classified by similar structure. How are they classified? We classify a molecule as a lipid because of what? Do y'all remember? What makes something a lipid?
Does it have something to do with like the glycerol bond?
Not exactly. It has something to do with its solubility. What it is and is not soluble in. What can you say about the solubility of lipids? Are lipids soluble in water or are they soluble in non-polar solvents?
Is it non-polar?
Right, they're not soluble in water. Madison said it, they're soluble in non-polar solvents. That's what makes something a lipid. But the body, what do we know about the body? The environment internally of the body is mostly water. It's aqueous. So how do we mix these lipids into our body? They're just going to want to clump together. Okay, imagine like butter and water. They just don't mix. So how do we digest these? This is a kind of a more complex process than it is with carbohydrates. Carbohydrates is going to seem a little more straightforward compared to lipids, if you can even call that straightforward. So, but we're going to look at digestion. Then we'll look at fatty acid oxidation. This is the process through which we can contain energy and convert lipids to acetyl-CoA. Okay. All right. Let me zoom back out. Digestion of triacylglycerol. So the focus today, even though there are other kinds of lipids, waxes and sphingomyelins and steroids and the others that I listed, we're going to focus on triacylglycerols because these are the primary source of biochemical energy in the body. Can y'all hear my light making that noise? No? Okay. It's making the weirdest, most annoying noise right now. Okay, triacylglycerol. So remember what a triacylglycerol is. I put a picture right here. It's a molecule of glycerol, that's the backbone. And then we have up to 3 fatty acids on a triacylglycerol, 3 fatty acids. So here's glycerol. You got the 3 carbons and the 3 OH groups, and then the pink is going to represent a fatty acid. All right. So if you have 1 fatty acid bonded to your glycerol in place of the OH group, that's a monoacylglycerol. If you add another one, that's a diacylglycerol, di meaning 2. And so we know triacylglycerol would be all 3 of those OH groups replaced with fatty acids. Okay, remember that's a tris, and they can be wildly different. I really should have put the table on here, but do you guys remember the table that I put on your resource page for exam 2 that had all the different shorthand notation for fats, stearic acid, lauric acid, palmitic acid, oleic acid, all those different fats? We're going to have to refamiliarize ourselves with some of those things. I'll put that table on your exam 4. Let me see if I can find what table I'm talking about really quick. That was chapter 23, wasn't it? Chapter 23 is right here. This table, that's the table that will be on a resources page for exam 4, okay? So we're gonna be using these words again: lauric, palmitic, stearic, oleic acid, linolenic, all of those, okay? So here's a nice graph from your textbook, and this just walks you through the digestion of triacylglycerol. So first, obviously the mouth and the stomach play their part, but that's really, really incomplete. That's just very preliminary digestion. They're still fairly undigested. They're just in their own little droplets. They don't mix at this point with water. So if you guys remember, we said there are bile salts, okay, that contain different sterols, like cholic acid or cholate in its anion form. And these are do y'all remember what we called them? We said they're something agents. We said bile salts act as
binding agents.
Close. They are going to bind, Cameron, but we had a specific name, emulsifying agents. Okay, so it's really once they get into the small intestine that bile acids, and then you also have some enzymes that go to work and they begin to really break apart different pieces of the triacylglycerols and emulsify them. Okay, these bile acids we said are emulsifying agents. So they help non-polar molecules mix in polar environments. So how does that make sense? Well, it makes a lot of sense if you look at this molecule. Everything in blue is very hydrophilic, polar groups. You see all these OH groups on the bottom. I'm going to circle them. And then we have the COO minus, this carboxylic acid group that's lost its acidic hydrogen. So it's all hydrophilic. This will mix with a polar environment like water. Then we have this hydrophobic side. These are what will mix with the fat, okay, and help digest. If you mix the bile salts and the fats together, it will cause everything to be able to mix because you have this molecule that has both a hydrophobic and a hydrophilic side, so it can mix with fats and with water, okay? So that's just kind of what this statement is saying right here. Separation of the hydrophilic groups, this is what allows fats, triacylglycerols, fatty acids, lipids to be emulsified. All right. So that's for fats that are really, really insoluble. Some fats are decently soluble, okay? Fats that are decently soluble, glycerol is a portion of a fat that's pretty soluble because it has all those OH groups on it. And then soluble fats directly enter the bloodstream after the small intestine. Insoluble fats have a lot more steps, okay? And we're not going to go into all the steps, but just know that fats are different. Obviously, lipids are different based on their structure. And we know that their structure is widely varied, okay? But the more insoluble, acylglycerols. I want to talk about this word for a second because you're going to see and hear this word acyl a lot. What does acyl mean? Why is it saying an acylglycerol? Anybody?
Is it a fat with an acetyl group?
Very close. You're gonna hear acyl-CoA a lot later, and that's very similar to acetyl-CoA. In fact, I have a figure that differentiates. There's a carbonyl group in acyl, but then so like if this were CoA or bonded to an enzyme CoA, the acyl part means that we don't necessarily have a definite like group on the other side. It could be a lot of different things. It could be long chains of hydrocarbons. It could be a short chain of hydrocarbons. Now, if what's on the other side of this carbonyl group is simply a CH3, then this is an acetyl group. But if what's on the other side of that carbonyl carbon is like a long hydrocarbon chain or just a bunch of different stuff, we just group it all together. We say it's an acyl group. So the way we represent it is like this. We put an R there for the rest of the molecule. It could be, again, a chain of 10 hydrocarbons, 20 hydrocarbons, hydrocarbon chains with double bonds. We don't know, but it's just some kind of hydrocarbon group. And so we just call it an acyl group, okay? All right, so insoluble acylglycerols and fatty acids enter micelles. We've heard this word before. You had to talk about micelles on exam 2, or I don't remember which one it was. Okay, so micelles, we know are fatty acids that have a polar head and a hydrophilic or hydrophobic tail, and they group together kind of the way this looks right here. And you have the hydrophilic part on the outside and the hydrophobic part on the inside. All right, now this picture is not, it's got some other things involved too. You see like this big yellow thing, we're calling that a protein. We see these little yellow molecules, cholesterols involved in I'm not going to call this a bilayer, but the membrane type thing here. And so we call this technically a lipoprotein because there's lipids. That's these little, you can see these little individual with the blue and the yellow tails that are making up the wall here. Those are lipids. But then we have proteins, protein complexes that surround the hydrophilic lipids, okay? And so that you got proteins. So we call these lipoproteins. And these are really, really important for the digestion of really insoluble lipids, okay? So they help transport them to our tissues, our adipose tissues, our immune system, where they can be further digested. So lipoproteins, once we dissolve things with emulsifying agents in the small intestine, we have pancreatic lipases. A lipase is an enzyme that digests lipids. Then they're packed in things like lipoproteins, micelles, and they're transported elsewhere where they're further degraded and further digested. Okay, so that's all I'm going to get into as far as the digestion of triacylglycerols. You can follow these steps down, but I haven't defined what a chylomicron is. It's just kind of a transport vehicle for triacylglycerols. They go to your lymph system, and then ultimately they get into the bloodstream, okay? Or they're stored in adipose tissue. So that's kind of an overview for the digestion of triacylglycerols. What's important to me here is that you remember what a triacylglycerol is, and that you remember how bile acids work, or bile salts, I should say. And then cholate is the example I'm giving here. You remember that we call it an emulsifying agent, all right? And that we have certain enzymes that go to work, and it mainly happens, here's what's really important, mainly all this happens in the small intestine. There's some preliminary digestion in the mouth and the stomach, but mainly all this happens in the small intestine. And then sometimes fats are dumped in the bloodstream from there if they're relatively soluble. Sometimes they're packed into lipoproteins, shipped throughout the rest of the body where they're further degraded and digested elsewhere. Okay. Questions on that? Okay, we're going to talk a little bit more about lipoproteins and lipid transport before we move on. So real quick, all fats come from one of three sources. What we eat, what we put into the body can contain fat molecules, lipids. Our adipose tissue already contains a store of lipids, or the liver contains lipids in progress, okay? There's a lot of digestion and things going on in the liver as well. And then as lipids are less dense than proteins, specifically lipoproteins, which again, kind of, this is what you're seeing here, this picture, this is technically a lipoprotein sort of vesicle. We classify them by density, okay? According to their lipid protein ratio. So I'm just talking about lipoproteins really quick. They're important for the transport of triacylglycerols, and they're transported different ways, okay? And so some lipoproteins take fats and they send them throughout the body. Some lipoproteins pick up fats throughout the body and they bring them back to the liver, okay? And that's what we're about to talk about. This figure here, I think, does a good job of explaining some of this in detail. So we've got lipids in the diet, we've got fatty acids from our adipose tissue, we have triacylglycerols synthesized in the liver, we have cholesterol, we know is a type of lipids synthesized in the liver, and then we also have cells that are continuously being renewed. We have dead cells that the body breaks down and gets rid of, and then we make new cells, okay? So we have cholesterol from dead cells. So how are all these different lipids managed in the body? How do we get rid of them if we need to get rid of them? How do we take them where they need to be? We have to have transport vehicles. Why? Because lipids, remember, are primarily hydrophobic. So they don't just mix into the bloodstream. They have to be packaged in a transport molecule. And lipoproteins are our main types of transport molecules. We just have different kinds. So you can see what each of these different lipids are transported by. Serum, I'm going to ignore chylomicrons. I'm going to ignore that one. But serum albumin, VLDLs, LDLs, and HDLs. Okay, now we're probably getting into some language that you maybe have heard before. So these are serum albumin, VLDLs, LDLs, and HDLs. These are all different kinds of lipoproteins that transport triacylglycerols around the body. So let's talk about each one real quick. Albumin, this is a protein. It's mainly, if you look the structure up, a lot of helix, you'll see alpha helices. This makes fatty acids water soluble. What's cool is it can bind one albumin molecule protein, and it's not massively huge, but it can bind up to 10 fatty acids per protein molecule. So it can really help emulsify a lot of different, a large number of fatty acids. What do VLDL stand for? Very low density lipoproteins. Okay, LDLs, low density lipoproteins. These transport cholesterol from the liver to the tissues. Is that healthy for our body to transport things, cholesterol, into our tissues? Do we want a lot of cholesterol into our tissues? Think about that. High density lipoproteins, that's HDL, transports cholesterol from our tissues back to our liver, where our liver can degrade it, get rid of it, break it down, and use the metabolites to synthesize other things. So if we're pulling the cholesterol content from our tissues and lowering the cholesterol levels throughout our body, is that a good thing? Okay, this is where we come up with good cholesterol, bad cholesterol. Okay, which kind of cholesterol is known as good? Which one's known as bad? Well, the good is the HDL. And then the bad is the LDL. Okay, again, what is HDL? What is it? These are lipoproteins that just have different densities. Okay, they're literally just classified according to their density. So the high density lipoproteins, these have a higher content of proteins in them. We said right up here, I'm going to put a star by this. Lipids are less dense than proteins. So the more lipid content of the transport vehicle, the lipoprotein, the more bad it is. Okay. The more it behaves like an LDL or a VLDL. The more proteins there are in your transport molecule, the higher the density, okay? Because proteins are more dense than lipids. So the more protein content you have in your transport vehicle, and this is what I'm calling the transport vehicle, it looks like that, then the healthier it is for you, okay? The higher the density and the more it takes cholesterol out of our tissues, brings it back to our liver. That's good for our bodies. So that's why doctors are constantly measuring your HDL versus your LDL levels and helping people that way if they really get in trouble with their cholesterol content. Okay. Questions on this? So what do I want you to know? I want you to know the difference between HDL and LDL. I want you to know what that stands for. And then how they're different. Like an HDL is going to have more protein content, and LDL is going to have more lipid content. And what they do, LDLs, they take cholesterol and they disperse it throughout the body into our tissues. HDLs, they take cholesterol from our tissues, they bring it back to the liver, and that's healthier for our bodies. Okay. I do want you to know, if I ask you a question about albumin, just that it's a type of a lipoprotein and that it helps emulsify and make fatty acids water soluble. Okay, that's enough about lipid transport throughout the body. We're now going to shift gears and talk about metabolism. Triacylglycerol metabolism. So we have this figure here, and trying to think where I want to jump in, I guess at the top. But if we are eating some food that has, fat in it, and so our body sees the macronutrient as lipids, okay? So the triacylglycerols in that food, how do we metabolize those? Well, the first thing we're going to do is we're going to hydrolyze, break it apart into fatty acids and glycerol, okay? This was one of the questions on exam 2. I said, and it was an optional one. Some people did it and some people did the sugar option. I want to pull it up to show you real quick what I'm talking about. So you actually are familiar with this. Go to exam 2. Right here. So remember we said hydrolyze a triacylglycerol containing lauric acid, palmitic acid. So I walk through this. So if you're confused on what that is, revisit this process. This is what's happening in our bodies when we begin to metabolize triacylglycerols. The first thing we're going to do is we're going to break the bonds right here, and we're going to break glycerol apart from the fatty acids. Okay, that's the first thing we're going to do. So I'm not going to redraw that whole picture, but I wanted to just show you that. So we're going to hydrolyze fatty acids and glycerol by lipoprotein lipase. Whoops, I'm on a lipase. So this is a type of enzyme that is specific for hydrolysis of triacylglycerols. Okay, so know what a lipase is. And then in chylomicrons, these are just a particular type of transport chauffeur molecule for fatty acids. Okay. If energy levels are good, so let's say we keep saying triacylglycerols, we can extract a lot of energy from them, but if we have enough ATP in our body, we're good on the energy levels. Maybe we've been running glycolysis, maybe we have good healthy glucose levels in our blood, we don't need extra energy, then we're going to store the fatty acids as, I'm going to abbreviate, triacylglycerols in adipose tissue. So we'll simply put the fatty acids and the glycerol back together and say, go to storage. We've got enough energy right now. We don't need to do any further metabolism. If energy levels are low, then we will continue with metabolism via beta oxidation. Okay. So again, it's always a catch-22 in the body. It takes a continuous assessment of what the conditions are, what the body needs, and things respond according to that. We don't just say, oh, we ate some fats and we're going to digest them. We're going to run beta oxidation every time. No, it depends. It depends on what we need in the moment. Okay, so this is what this figure is showing though. The first thing, so again, the brown arrows are catabolism, the breaking down of molecules. The blue arrows are anabolism, the building up, okay, putting things together to make other things. So the first thing we will do is hydrolysis, and that gives us our individual fatty acids plus glycerol, breaks it apart. And then if we need energy, we'll go ahead and run this, beta oxidation. If we don't need energy, then we won't run this, and we'll just come back up this way and we'll store it as a triacylglycerol. Okay. Now, glycerol, remember from yesterday, this was the very, very last slide we went over, but glycerol can enter glycolysis or gluconeogenesis. Okay. I'm going to jump over to that slide really quick. Do you guys remember the very, very bottom? We said right here, glycerol can be converted to DHAP. This is an intermediate in glycolysis, and it can jump right into the glycolysis pathway if we need it. So there's a lot of versatility right there as well. And this is one way that lipid metabolism is connected to carbohydrate metabolism. I may ask you that on the exam. Tell me how these pathways are connected. Well, glycerol can be a direct intermediate into, or it can be metabolized to an intermediate in glycolysis. Okay. Or triacylglycerols can be metabolized to acetyl-CoA and jump right into the citric acid cycle. All right, let's go back. So I just wanted to point that connection out, and that's just what this green arrow is showing you right here on the figure. Okay, so you see the end result of beta oxidation is acetyl-CoA. Okay, we are very familiar with this molecule now, and then what happens with acetyl-CoA is the citric acid cycle, or that's the green arrow. It can go to the citric acid cycle. Or we talked about ketone bodies yesterday. We're going to come back and talk more about ketone bodies today. But we did introduce that yesterday. Okay. Adipose triacylglycerols only happen when, so hydrolyzed fatty acids transported by linked to muscle cells, liver cells, converted to acetyl-CoA. I don't remember why I put this bottom part on here. Sorry. Oh, albumin. Okay. Hydrolyzed fatty acids and glycerol by lipases, transported by albumin to muscle and liver cells, converted to acetyl-CoA. So this is when energy is low, and we do want to move forward with metabolism. So we do the hydrolysis first, and then we have to transport it to the liver, and then we're going to really metabolize it with beta oxidation. Okay. Any questions on this figure and kind of the pathways that you're seeing? All right, so now we're going to ramp up to, we're going to really get into a few more details of metabolism. Okay, so remember I said glycerol can be converted to DHAP and then can go straight into gluconeogenesis or glycolysis. We're going to look at how that happens really quick. Like, what are the steps that convert it? So glycerol from triacylglycerols produced by the hydrolysis of fatty acids. So let's say we have a lipase and we do hydrolyze our fatty acid, and we have glycerol now. So there's a two-step process to convert it into DHAP, the dihydroxyacetone phosphate. And this is produced, does anybody remember what step in glycolysis produces DHAP? It was one of the energy investment steps. Produces DHAP. We go back up to glycolysis.
It wasn't 6, was it?
Very close to 6 is step 4. So again, sorry, this figure, I have another figure for y'all on glycolysis. I'm going to share it after lecture day. It's a little not so blurry when you zoom in. But fructose 1,6 bisphosphate, when we cleave it right down the middle and get our 2 products, one of those was DHAP. And then that's immediately isomerized to glyceraldehyde 3-phosphate. Okay, so technically step 4, and then it's converted to glyceraldehyde 3-phosphate in step 5. Okay, so we're about to see how glycerol can be converted to DHAP. And glycerol is synthesized or it's produced when we hydrolyze fatty acids. So there's glycerol. The first step is we have to couple this with ATP hydrolysis. So that's what you see there. We have ATP, we're going to hydrolyze it, and we're going to put the phosphate group right here on this 3rd carbon. So there's our phosphate group from ATP. Claire asked me this question yesterday. This is part of a homework question. Like, why do, metabolism is all about generating energy in the body. But oftentimes we see that there are these steps initially where we invest energy, like a small amount of energy. Why do we do that? Why does the body do that? We're trying to make energy, but it costs us some energy on the front end. Why do you think we do this? Why is this necessary to like phosphorylate something?
Is it like the coupling reaction where it's endergonic and needs an exergonic?
That's one reason. Maybe the reaction is simply endergonic on its own. And so coupling it to a larger exergonic reaction simply allows the reaction to happen. That's one reason. Claire and I also talked about something else. So there's 3 reasons in my head that I'm thinking of. That's one. A lot of times our initial starting material is already very stable. And to break it down via metabolism, we kind of have to destabilize it a little bit. And we can do that by adding on these high energy groups. Okay, ATP is we call it a high energy molecule. But when we phosphorylate something, we're in part destabilizing our initial substrate so that we can then either cleave it or break it down or send it to a pathway that can further metabolism. And then another reason we add energy or do things with ATP is so that certain steps are irreversible. The body doesn't like to take ADP and make ATP. It's got to have a lot of energy to do that. So there's a specific way we do that. That's oxidative phosphorylation. But we run it couple it with a reaction like the hydrolysis of ATP so that things are irreversible. One thing's like glucose. Why do we phosphorylate glucose? That's the first step of glycolysis, so that it doesn't just diffuse right back out of the cell. Okay, so it stays put, and that process kind of commits it towards the pathway of glycolysis. Anyways, I wanted to bring that up because that is a homework question. Okay. But anyway, so coming back to this, we have glycerol 3-phosphate now. So we still have glycerol, we just have a phosphate group on carbon 3. One more step, we take NAD+, reduce it to NADH, and now we have dihydroxyacetone phosphate. So we just take off these hydrogens right here on carbon 2 and make it a double bond. Now we have dihydroxyacetone. Very simple. And this is an intermediate. And glycolysis. So we just looked at that. It's made in step 4, converted to glyceraldehyde 3-phosphate in step 5, and you can run glycolysis. So these are all the different scenarios or the different pathways that can arise from making DHAP. So we said it can enter glycolysis. From there, we're going to produce energy. It can enter gluconeogenesis, so we'll make glucose. So why would we need to do that? Well, let's say our blood concentration of glucose, our glucose concentration in our blood is low. Well, if we need to bring that back up, we could simply go through gluconeogenesis, and instead of running glycolysis, we can just increase the concentration of glucose. So we could make glucose that way, or it can be formed back into triacylglycerols. So, and we can store it for energy, okay? So we could remake glycerol if we needed to. There's specific enzymes for that, and we're not gonna look at that pathway. So the main reason I'm showing you this is because I want you to see how interrelated lipid metabolism can be to carbohydrate metabolism. And it's through this connection right here, glycerol and DHAP. Questions? So this is how, or this is one way, lipid connected or linked to carbohydrate metabolism. All right. Okay. We're almost to beta oxidation. One more slide and then we'll be there. And I want to bring you back to this idea of the fact that metabolism is a very finicky thing. It's very temperamental on, the body's always taking an assessment of what it needs, and then things respond and react according to our current needs. Okay, so we have to decide when we eat food or when we're digesting lipids, do we need to store it or do we need to mobilize it for metabolism? Okay, mobilization just means get it out of the adipose tissue and get it ready for beta oxidation. Okay, so remember, we store triacylglycerols in adipose tissue. And this is the process for that you see here, storage. The opposite of that is metabolism. Get it out of storage and let's go break it down for energy. So they're kind of the opposite processes. So let's look at this figure. Glycerol 3-phosphate. We just looked at glycerol 3-phosphate on the previous slide right here. That's just glycerol with a phosphate group on carbon 3. To store this, if we need to store lipids, because we don't need energy right now, then there's a couple of steps. These are called acyl transfers. These are what we call these reactions. They're run by transferase enzymes, okay? Acyl transferase. So the way this looks is this is the acyl group. This is what I drew earlier. We got the carbonyl and oxygen, and the R just stands for some long hydrocarbon chain. It could be any kind of fatty acid, okay? And you see it bonded to what? CoA, and coenzyme A, okay? So if we're storing it, we'll take it off of coenzyme A and we'll put it onto glycerol. That's the acyl transfer. We transfer it from coenzyme A to glycerol 3-phosphate. All right, so this is the first transfer. Now it is bonded technically to that top carbon in glycerol. Then we do it again, another acyl transfer by a transferase enzyme. So here would be the next fatty acid. We take it off, we transfer it from CoA to the next spot on glycerol 3-phosphate. The end product here is known as phosphatidic acid. All right, and then we could do one more to get a full triacylglycerol. All right, so 3 full acyl transfers to get a triacylglycerol. We store that. All right, and that's what you see in the bottom. We get the end product is a full triacylglycerol, but we take phosphatidic acid. We have to get the phosphate group off. Now the phosphate group that was here is now an OH group. Now we can do one more acyl transfer. Okay. So it's a multi-step process, but we're just transferring these acyl groups back onto glycerol to make our full triacylglycerol. We can store that. And these are handled by transferase enzymes. Okay. All right, so that's storage. If we have plenty of energy in the body, we don't need to metabolize lipids. But what if we do need to metabolize lipids? Okay. So long after a meal, when we eat a meal, our body gets a lot of stuff and it decides what it needs to do. But long after we eat it, after we've digested it, after things have kind of restored equilibrium in our blood and our tissues and our cells, long after a meal, glucagon insulin, kind of backwards. Insulin levels drop while glucagon levels rise. And these changing hormone levels activate a lipase called triacylglycerol lipase. Okay. So we talked about insulin and glucagon yesterday. Do you guys remember kind of just the summary of how these two hormones work? Insulin and glucagon.
Insulin make your blood sugar go down. Glucagon make your blood sugar go up.
Thank you for that summary, Robin. So glucagon dumps glucose into the blood, insulin pulls glucose out. Okay, so these are these are the regulatory molecules for glucose levels, but we see here that they're also stimulating the mobilization of triacylglycerol, so lipid metabolism. Okay, so if we have low levels of glucose in the body long after a meal, maybe we're kind of deplenishing our glucose supply, which is deplenishing our energy supply, but we haven't eaten anything else yet. We're not eating anything. We're not taking in any new nutrients or fuel. Then our body starts to say, okay, maybe we need to start pulling some lipids and triacylglycerols out of storage and metabolizing those for energy. Okay. So these changing hormone levels activate this enzyme, triacylglycerol lipase, and that is what's going to start the breakdown of triacylglycerols. Okay. So here's kind of the summaries of what we've talked about. Is energy demands are not being met by glycolysis, fatty acids and glycerol are dumped into the blood and sent to generate energy. But if energy demand is met or very low, fatty acids and glycerol are resynthesized back to triacylglycerols for storage, and that's what we walked through here. So again, all this metabolism, it just depends on what the body needs in the moment. All right, now we're getting to actually my favorite part of the lecture. So we talked through this earlier. What's the difference between acyl and acetyl? And I have the pictures there to remind you. So there's kind of before we get to beta oxidation, which is really the big key thing for today. It's the main pathway that I want you to know. There's like a pre-step, okay? So first, maybe we've activated triacylglycerol lipase, and now we have some fats that are some triacylglycerols that are getting ready to be metabolized. Well, the first pre-step that we have to do is we have to take our fatty acid and we have to form fatty acyl CoA. We're going to link it up to an enzyme, a coenzyme A. We're going to link the fatty acyl, you might call it a fatty acid, fatty acyl, fatty acid, kind of the same terminology there. We're going to take the fatty acid. It was bonded to glycerol, but now we've hydrolyzed it and we just have an individual fatty acid. So this could be stearic acid, oleic acid, lauric, palmitic acid, any one of those fatty acids. And we're simply going to take coenzyme A. We're going to have to use ATP. So there is an investment step here. We have to invest ATP, or an energy investment step, I should say. And we're going to form fatty acyl CoA. Now, if you notice, it says the product is AMP. What is AMP?
Oh, was that the mono?
Yeah. So if ATP is this, and then you got the phosphate group, phosphate group, phosphate group. This stands for the adenosine. AMP would just be one phosphate group. We've actually broken off two phosphate groups. So later on, when we are calculating how much energy we get, we actually say it's like it required 2 ATP molecules. Okay, and I don't want that to be confusing because it actually doesn't have a 2 right here, but we take off 2 phosphates. So we hydrolyze it twice. So later on, when we're doing our final energy calculations, we're gonna count that this caused for 2 hydrolysis, we say 2 ATP. Okay, so I just wanted to introduce that here. We'll come back to that later. Okay, so this is our pre-step. We've now bonded this fatty acid or fatty acyl to coenzyme A. Then we just have to get it to the matrix, because we know in the matrix of the mitochondria is where citric acid cycle and oxidative phosphorylation happens. And the end product is going to be acetyl CoA. So we've got to be in the matrix so that can feed right into the citric acid cycle. Okay, so I want to ignore all this part about don't worry about that. I just want you to know that we activate it and then we transport it to the matrix. Okay, so if we're committing to the metabolism of a triacylglycerol, step 1 is we hydrolyze it from glucose. Step 2 is we activate it. We activate it by joining it to coenzyme A. Step 3 is we move it to the matrix. Okay. All right. So then we're ready for beta oxidation. Okay. And the whole pathway is right here on the next slide. And we're going to walk through it step by step. But I'm going to tell you 2 things about beta oxidation. First off, why is it called beta oxidation? When have we talked about Greek letters before in this class? You guys remember? Alpha, beta. Well, we talked about it with carbohydrates, whether the OH group points up or down above the ring, but that's not the context here. We don't have a carbohydrate. We talked about them when we were naming carboxylic acids. You guys remember? And we were naming carboxylic acids. We talked about how the different carbons connected to or next door neighbors to the carbonyl carbon can be called alpha and beta. So that's what you see here. If we're looking at a fatty acyl CoA, so I don't know what fatty acid is. We don't know how long this extends. We have an R there. So the molecule extends the rest of the way that way. But we have coenzyme A here, connected through this sulfur. Here's our carbonyl carbon. I'm going to highlight this, carbonyl carbon. So then we would call this carbon the alpha carbon, and we would call this carbon the beta carbon. So this was all the way back in chapter 17. We called this the common naming system. There was the systematic and the common naming system. In the common naming system, they would use the Greek letters. Okay, so we're bringing this back here. That's where this whole pathway gets its name from, because we're about to heavily manipulate this beta carbon. Okay, so what we're going to derive from beta oxidation is acetyl-CoA. So I'm going to draw that really quick. Acetyl-CoA. Draw it with me. Okay, so I want you to look at this fatty acid acyl-CoA, and I want you to look at acetyl-CoA. This is AC. And I want you to see kind of how they're similar. See how they are similar. So the parts that are similar, I'm going to highlight. Sorry, it's super loud up there. The parts that are similar is all of this right here. Okay, and then we're going to have one other CH3 group attached. Okay, so keep that in mind. That's going to be the final product of acetyl-CoA. And the way this pathway works is kind of like this. So we've talked about metabolic pathways. And we said in a pathway that when you run a reaction, the product of one reaction is often the reactant for the next reaction. And that's how pathway works. You run a pathway, you get a product, and then that's the reactant for the next thing. But there's different kinds, okay? There can be a straight chain pathway where you have steps 1 and then steps 2 and then steps 3, so on and so forth, kind of a straight unbranched pathway. But the citric acid cycle is actually a circular one, right? Where the final product, we know oxaloacetate formed in step 8 is actually the reactant for step 1. Okay, so there's straight chain pathways, there's circular pathways, and we see something different here. We see this is called a spiral pathway. So the way this works is we're going to run a series of reactions, 4 steps. It's going to have 4 different enzymes. So we're going to take our initial reactant, we'll call it A, substance A, and we're going to run it through this chain of reactions. It's a straight path, 4 different enzymes, boom, 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 boom. And at the end of every single thing, we're going to get acetyl-CoA. Okay, so the only way I want to manipulate this figure, I pulled this straight from your textbook. We don't have to run all of this before we get the final product. We get the final product after every series, but it's just that it happens again and again and again. So A will then become B. It'll be shortened by 2 carbons, okay? We will have taken this carbon and this carbon off of the fatty acid. Now that's on acetyl-CoA. And so our starting material will be shorter by 2 carbons, and that will give us B. Then we'll run the chain again, beta oxidation again, get another molecule of acetyl-CoA, and our fatty acid will again shorten by 2 carbons every time, okay? And we just do it again, again, again until the whole fatty acid is broken down. Okay, I'm writing that in there. Shortened by 2 carbons each time. Where do those 2 carbons go? They're in the acetyl group of acetyl-CoA. So we just break off 2 carbons, put it on acetyl-CoA. Break off 2 carbons, put it on acetyl-CoA. Break off 2 carbons, put it on acetyl-CoA until the whole fatty acid, it might be 10, it might be 20 carbons long, is metabolized. All right, so that's what we're going to look at now. Beta oxidation. So this is in 4 steps. We're going to walk through one at a time. So I'm going to zoom in on the first step. The first step is what we call a beta oxidation step. So we're going to take the beta carbon and we're going to oxidize it. We're going to take the beta carbon and we're going to oxidize it. The enzyme that does this is a dehydrogenase. Acyl-CoA dehydrogenase. And we're familiar with these. These are the enzymes that were the major component of the citric acid cycle. They were involved in several steps of the citric acid cycle. So same idea that you saw with the citric acid cycle in the steps where we would make carbon dioxide and we would reduce a coenzyme, that was a dehydrogenase, same type of enzyme here. So 2 hydrogens are going to be removed. And then we're also, this is going to involve FAD, the coenzyme FAD. We know it's a dehydrogenase because it involves a coenzyme. FAD is going to be reduced to FADH2. So let's look at this. So let's say that we have some fatty acid and we're calling this small, it's been activated, it's been linked up to coenzyme A. So we call this whole complex a fatty acyl-CoA. Here is my fatty acid part. And we see this little N right here because we don't know how many CH2s. This could be real. It could be any number of fatty acids, okay? But I want you to focus on the carbons that are labeled beta and alpha, okay? So those are labeled for you, oops, right here. All right, so we're gonna take FAD, the dehydrogenase is gonna do its magic, and what we end up with, we are essentially gonna take a hydrogen, one off the alpha and one off the beta carbon. One off the alpha, one off the beta carbon, and we're going to end up with, I'm going to highlight the portion that's important right here, a double bond now between the alpha and beta carbon. So the name of the enzyme makes sense. We are hydrogenating. Okay, we're taking hydrogens off. So we're moving hydrogens, so dehydrogenase. Okay, now whether you know the names of these is not so important to me, but I do want you to be able to walk through this. If I gave you, in fact, this is a homework problem. This is one of your homework problems that you can write the product of the first and the second and the third and the fourth step of beta oxidation, okay? So the first thing is that we take a hydrogen off the beta and the alpha carbon, we make the double bond. And in the process of this, we've reduced FAD to FADH2. All right, questions on that first step and what happened? All right, step number 2 is a hydration step. So this is performed by a hydratase, which we also saw in the citric acid cycle whenever you're adding water to something. So OH is going to be added to the beta carbon. So we have oxidized the beta carbon. Now we're going to further oxidize it. We removed a carbon-hydrogen bond. Now we're going to make a carbon-oxygen bond. So this was the same thing we ended with in step 1. Here's the double bond. Now we're going to take the hydratase. Here's water. We're adding water. And here's the OH group from water. And here's the H. So we break water apart, H and OH. And the OH is going to bind to the beta carbon. And the H is going to bind to the alpha carbon. Okay. So I guess my picture over here, I should circle, this would be like the hydrogen from water. So it's the hydratase that does this. So we've broken the double bond and we just put the OH on the beta carbon and the hydrogen from the water goes on the alpha carbon. And this is further oxidation. Why? Because we now have one little carbon oxygen bond here. Remember, oxidation can be the removal of hydrogen. It could be the loss of electrons. It can be the increase of a carbon oxygen bond, increasing the number of carbon oxygen bonds. So this is all oxidation. All right, step 3. This is called the second beta oxidation. It's another dehydrogenase. So now the beta carbon is going to be converted from an alcohol. Why are we calling it an alcohol? Because it just has this OH group bonded to it. That's an alcohol functional group, right? So we're going to convert it from an alcohol to a carbonyl, carbon oxygen double bond. So here is our alcohol. There's the beta carbon. We're going to remove this hydrogen and this hydrogen, both off the beta carbon. So this is, again, a dehydrogenase. And in the process, we're going to reduce NAD plus to NADH. We're going to reduce NAD plus to NADH. All right, so now here's the new look to our molecule. This carbonyl in blue that I'm going to highlight, this never changes. This stays the same the whole time. This is always there. Okay, that part never changes. Okay. All right, so that's step 3. Step 4 is the last part. Now we're about to get acetyl-CoA. So we're going to cleave the alpha beta carbon bond. We're going to cleave the bond between the alpha and the beta carbon. Okay, this is a thiolase enzyme that does this. Thiolase. So the alpha carbon beta carbon bond is going to be broken. The acyl group is going to be attached to another CoA. And in the process, one acetyl-CoA is going to be produced. So let's look at this very closely because this is a lot happening in one step. So come over here to our picture. So this right here is the bond we're going to break. And then I'm going to highlight the acetyl-CoA. When we break that bond, look. This is acetyl-CoA. Now we have to add another little hydrogen. A little hydrogen is going to be added to this carbon to make it a CH3. But that's acetyl-CoA. So we're going to break that bond and we form acetyl-CoA when we break that. Okay, but then this whole acyl group that I'm going to highlight in red, this is going to be left without a coenzyme A. It's like it's naked. It needs to be bonded to CoA. Okay, it needs to be activated again for the next run through of beta oxidation. We can't leave it without coenzyme A. So we have another molecule of coenzyme A. We have to have this little thiol group right here, which is why it's called thiolase. Because all we're simply going to do is we're going to link these together, and then we're going to link this together right there. So we reform. This is the fatty acyl-CoA. It's just 2 carbons shorter. Those first 2 carbons are cleaved off, the alpha and the beta. Or sorry, the alpha and the original carbonyl carbon. And that is what, so the ones in yellow are right here on acetyl-CoA now. And the ones that were in red, these are, so this was the beta carbon. Now it's the prime original carbonyl carbon. And we can assign a new alpha and a new beta for the next run through of the whole pathway. And this is how we make acetyl-CoA from the metabolism of fatty acids. Okay, so for the exam, we're not done yet, but for the exam, I want you to know these 4 steps. Now, I'm not going to drill you on what's the name. So here, the names aren't so important to me, knowing all the names of these. If that helps you, by all means. use the names to help you remember where you are. But what matters to me is that you know these 4 steps and what's happening, and that you could, if necessary, draw these out. So there is a homework problem. We're going to do we're not going to work an example right now, but we are going to look at how much energy we get from this pathway in a minute. We'll do a calculation for that. I want you to know what enzymes are involved, whether it's a dehydrogenase or the hydratase or a thiolase. Oh, and I want you to know where coenzymes and what coenzymes. I'm going to highlight those in pink. FAD and NAD. We reduce FAD in step 1 to FADH2. We reduce NAD to NADH and another H plus in step 3. Okay. So here's kind of the overall summary from beta oxidation. Because we're about to look at how much energy. We say this creates a lot of energy. Well, how much? The overall summary points. Beta oxidation breaks down fatty acids 2 carbons at a time. Now, in the very last cycle, you're actually going to get 2 acetyl-CoAs. Let me prove that to you. Let's say we have let's say we have a carbon or fatty acid that is only 1, 2, 3, 4, 5, 6 carbons long. Okay. Well, after 1 run of beta oxidation, if we run beta oxidation 1 time, what are we going to be left with? Our product will be shortened by 2 carbons. So 1, 2, 3, 4. Okay. And we get a coenzyme. We get an acetyl-CoA plus the acetyl-CoA. Got this. Okay, so we get these 2 products. So you see we went from having 6 carbons. Now if you counted those, we only got 4. So in the last run, when we only have 4 carbons left and if we run beta oxidation, so here's 1, 2, 3, 4. If we run beta oxidation again, look what happens. See how our products are identical? Because we're breaking it off. This would be the 2 would be the alpha, and that would be the beta, and we know we break it right there between the alpha and the beta. So we're going to get 2 identical acetyl-CoAs. So I wanted to make that point here on that. In the very last cycle of beta oxidation, we always get 2 identical products, 2 acetyl-CoAs. Okay, let's keep going. Another kind of overall way to summarize this: 1 FADH2 and 1 NADH are produced. And then remember, and I think I copied this down from the textbook when I wrote this lecture, so this should be 2.5. That 1 FADH makes 2 ATP, and this is, we're talking about electron transport chain here and oxidative phosphorylation. That's where we get that relationship from. And 1 NADH makes 2.5 ATP. Same thing. This is, we get this relationship from chapter 21 when we walk through the electron transport and oxidative phosphorylation. So we're going to need to remember those and use those in just a minute. Okay. Acetyl-CoA and the reduced coenzymes all feed into the citric acid cycle and electron transport to make ATP. All right. So up here, when we are looking at the products, we could literally write in this goes to electron transport chain. This goes directly 2 electron transport chains. So it's going to make 2.5 ATP here. And this would make 2 ATP here. So how many ATP do we just get from the coenzymes of beta oxidation? How many ATP do we just get from making these reduced coenzymes that can feed directly into electron transport? This is some really hard math, I know. 4.5, right? So remember that. Every cycle of beta oxidation produces 4.5 ATP. We're going to need that relationship in a minute. Okay, so 2 quick questions here because we are about to calculate. Let's say we have like stearic acid, or I think, what does it give us? We're going to use an example of a fatty acid, and we're going to calculate how many ATP do we get. So yesterday, you guys remember when we did glycolysis and we looked at like all the ATP that we make and the NADH, and we added up how many ATP do we get from 1 molecule of glucose? Do y'all remember? How many ATP do we get roughly? It's not a perfect system. How many ATP do we get from 1 molecule of glucose? Do y'all remember where we calculated that? Do y'all remember this slide? We added it all up. Y'all are so quiet today. We said 33 ATP from 1 molecule of glucose. If you look it up on the internet, it might be 38, depending on what relationship they use between NADH.
33 ATP.
Thank you. Okay, now we're about to take a fatty acid and say, how many ATP do we get? I think it's going to blow your mind, okay? So, how many ATP are generated from 1 molecule of acetyl-CoA? We figured this out, that it was 10.5. If you don't believe me, look at the citric acid cycle. This is a citric acid cycle. We get 1, 2, 3 NADH, and we get 1 FADH, okay? So 3 NADH is going to make 7.5 ATP because each one makes 2.5 ATP. And 1 FADH makes 2 ATP. And we said we already get 1 more ATP just from the cycle itself. Just from the cycle itself, we get 1 ATP. So if you add all that up, that was 10.5 ATP. Okay. So that's where I'm getting this number, 10.5 ATP, just from 1 molecule of acetyl-CoA. And then we actually just answered this. If the coenzymes, so the NADH and the FADH2, just from beta oxidation. Oh, yes, Cameron.
What did you abbreviate that CAC?
Citric acid cycle.
Oh, okay, my bad.
Sorry. If the coenzymes from beta oxidation pathway also feed into electron transport chain, how many ATP are made just from a cycle of beta oxidation? So we answered that. We said 2 ATP plus 2.5 ATP right here. I marked through it. Plus 2.5, that gives us 4.5. Okay, so I'm gonna write this down here. So we get 4.5 ATP just from 1 cycle of beta oxidation. I'm abbreviating again, beta oxidation. Okay, so let's take that information and let's apply it to a real fatty acid molecule. So there's a few steps here. This is probably the most math that we have done all summer in this whole course. This is the most math you're going to do in this whole course. Okay. So my math brain people get
Oh,
excited.
Question real quick.
Yeah.
Mine looks different. I have an added.
Oh, I did have to make an edit last night. Thanks for bringing that up. I think there might be like a +1 right here.
And then the 5 is supposed to be a 4.5.
Yeah, change this to 4.5. Make those 2 corrections real quick. I caught those last night when I was going over the slides. So just take off the +1 and change this to, just put a 4 point in front of the 5. Thanks for reminding me of that, Robin. All right, so here's the whole point of this slide. We're going to determine how many acetyl-CoA molecules are produced from a fatty acid, and then we're going to figure out how many ATPs we get overall. So remember that 2 carbons are removed from the fatty acid yield 1 acetyl-CoA every time you run beta oxidation, and then the last cycle yields 2 acetyl-CoA. So the easiest way to do this is you're going to look at your fatty acid. Let's just go pick a fatty acid. Right here. Which one do y'all want?
One you would put on the test.
I could put any of them. Hey, remember I said be nice to me today.
I think I was making my coffee.
When I said be nice to me?
I heard be nice, but I didn't know why. But I heard be nice.
Because my daughter, I only have 1 month left as a 2-year-old. She's about to be 3 and I'm not ready for it. Whoops. I'm gonna run through a simple one for y'all's sake. Well, I will totally be happy to do a more complicated one later. Let's just pick well, let's do stearic acid. Okay. So I'm gonna write the formula for stearic acid over here. So it was CH3, and then we had CH2, was it 16? COOH. All right, so step number 1, once you figure out what fatty acid you have, and I will always tell you either stearic acid or oleic acid or something from that chart. I'm not gonna pick something that's not on that chart. And you will have the chart as a resource page for the exam. Now, we want to figure out how many acetyl-CoA are we going to produce from this fatty acid. All you do is you take the total number of carbons in the fatty acid, you divide by 2. So do that with stearic acid. How many carbons are in stearic acid? Divide that by 2. That's how many acetyl-CoAs you'll get. So how many carbons? I was doing this last night and it was so late that I was thinking, oh, there's 3 carbons right here. No, that's hydrogens. I kept getting the wrong number and I was, what is wrong with me? So we have 1 carbon here. We've got 16 more right here, plus 1 more right here. So we get 18 carbons, right? So we're just gonna do some simple math. We're gonna do 18 divided by 2. We should get 9 acetyl-CoA. Now, number 2, using the relationship from the previous slide, so I'm going to highlight them. These relationships right here. Convert the number of acetyl-CoA to ATP. So, 1 molecule, marking through it. That's on 1 molecule of acetyl-CoA equals 10.5 ATP. From stearic acid, we're going to make 9 acetyl-CoA. How many ATP? So we do the number of acetyl-CoA right here. So we'll plug in a 9 right there. And we know for every 1 acetyl-CoA, it's 10.5 ATP. So do that math. to 94.5. Okay, so we would get a 94.5 ATP so far, and this is not. Do you see how much more ATP we get from breaking down fatty acids already than from one molecule of glucose? We're already got triple the amount, and we're not even done yet. Okay, step 3 is that we determine the total number of ATP produced just from beta oxidation itself. So we know every cycle of beta oxidation makes 4.5 ATP, but how many times are we going to have to run this fatty acid through beta oxidation? So figuring out the cycles of beta oxidation is relatively simple, but there's just a little trick you just have to remember or write down on your note card or something. It's going to be the total number of acetyl-CoA minus 1. Okay, because why is it minus 1? Why is it just not the same number of acetyl-CoA? Because remember, the last time we run beta oxidation, we get two acetyl-CoA. So for example, if we have stearic acid, I'm going to try to draw it really quick. 18 carbons, 2, 4, 6, 8, 10, 12, 14, 16, 18. Okay, there's stearic acid. So let's put a black little mark on our molecule. And this little black mark is going to represent one cycle of beta oxidation because we know it's going to break it apart every 2 carbons. So this would be carbon 1 and carbon 2, and then we're going to break the molecule apart. That first mark represents one cycle of beta oxidation, and then we break off that first acetyl-CoA. 2 carbons later, we're going to break it here. 2 carbons later, we're going to break it here. This is 3 cycles of beta oxidation. 4 cycles of beta oxidation. 5 cycles of beta oxidation. 6 cycles of beta oxidation. 7 cycles of beta oxidation. 8 cycles of beta oxidation. And we will have metabolized all the carbons. So do you see how we'll get 9 acetyl-CoA, but that's because this last cycle of beta oxidation right here will give us a molecule of acetyl-CoA from those 2 carbons and from those 2 carbons. So we make 9 acetyl-CoA, but we only have to run 8 sets of beta oxidation to break all of it down. So for stearic acid, this will be different depending on what fatty acid you're looking at. But for stearic acid, we're going to run 8 cycles of beta oxidation. Each cycle makes coenzymes, and those coenzymes go and produce 4.5 ATP. Have I lost anybody yet? Every time we run beta oxidation, remember step 1, we get F1 FADH2. And then in step 3, we get 1 NADH. And those go and make ATP. Together, total, they make 4.5 ATP. So we got to do some math here. 8 times 4.5. Don't grab your calculator. You don't need your calculator for this. 8 times 4 is 32. Half of 8 is 4. 32 and 4, so you should be 36. I want to do it in red. 36 ATP. Just from the coenzymes produced in beta oxidation. So we are already making 94.5 ATP from all the acetyl-CoA we make. We're going to make another 36 ATP just from the coenzymes that we happen to make as a byproduct of beta oxidation. Then remember, I said in the activation of the fatty acid, before we even transport it to the mitochondria and run beta oxidation, that we have to invest 2 ATP to just activate the thing. So we're going to subtract 2 ATP. So here's the total math that we're going to do. 94.5 plus 36 ATP minus 2. In total, That will give us what? Somebody double check me. You get 128.5.
I think I got 122.5. Let me check again. 94.5 plus 36 equals minus 2. Oh, yeah, I was wrong. Just kidding. Huh? See, I'm not good at math.
No.
Even with the calculator.
That doesn't mean you're not good at math. You got the right answer.
I just did it with a calculator. I had to do it twice to get the right answer.
That's okay. I told you last night I was trying to count hydrogens as carbons. Okay. Does this seem mind-blowingly hard?
Did you say
I don't
that
want to do it.
What, Abby?
Did you say we subtract the 2 ATP because that's what we used from the beginning?
Yeah, subtract 2 ATP that we invested in the activation of the fatty acid before we even run beta oxidation. So that was right up here. I said, remember, we're investing 2 ATP right here, so we got to take those out.
Okay, thank you.
Okay. You're going to have to do one of these on the exam.
Would there ever be a time where like you'd only want us to do one of these steps, or would it be like all 4 together?
Yeah, I mean, possibly on like a multiple choice or something, I could have you just calculate the number of acetyl-CoA. Or I could ask you, I could say, here's a fat, how many cycles of beta oxidation would you have to run on this fatty acid to completely metabolize it? Sure. Questions on that? Here's more practice problems, but we're kind of at time for the day. I kind of like to take a break. But here's what we have left. So before we go, we don't have a lab today or tomorrow's kind of nice. So there's more practice here with lauric acid and then with palmitic acid. And then what happens when we have too much acetyl-CoA and ketone bodies. And then I actually decided that I'm going to introduce lipogenesis, but I'm not going to get into all the steps of you knowing the biosynthesis of fatty acids. So we're probably not really going to dig into these last 2 slides or last 3 slides. We're probably just going to look at this letter here and be done. So, I mean, do y'all want to go ahead and finish these 2 slides and we can actually be done for the day? Or do y'all want to take a break and come back? For like
Yeah.
I want to go ahead and finish.
Go ahead and finish.
It's 2 slides.
2 slides.
Yeah.
Y'all down? Okay.
If I start glitching, then all right.
Okay, so I'm going to jump over the practice slide, but today in my office hour or whatever, I really think and I might introduce lecture tomorrow. I might start lecture tomorrow with these practice problems. Okay, we are going to do these before the exam because we need more practice together with this. One is not enough. Okay. So let me finish by saying, I hope this is astounding to you. It was astounding to me that by the breakdown of a triacylglycerol, and this is just one fatty acid, a triacylglycerol, remember, is your glycerol molecule with 1, 2, 3 fatty acids. We just looked at the breakdown of 1 fatty acid. So if I had a fatty acid with 3 steric, or if I had a triacylglycerol and all of these fatty acids were steric acid, then the breakdown of all 3 of those would produce triple the amount. Isn't that crazy? Like almost 400 ATP just from the breakdown of 1 triacylglycerol. Okay, so now you see why we say that the storage of glycogen and glucose in our body is less than 1% of our energy reserves. It's a teeny amount compared to the energy we can get from our fats. So Now, here's a question I want you to think about too. Write this down. I don't have it on my slides. If the metabolism of fats is so like, I guess beneficial energy wise, like we get so many ATP from it, why do we even need glycolysis? Like why is glycolysis the body's main way for getting ATP? We don't use this type of metabolism. We don't break down our fatty acids unless we just have no glucose in our bodies and we've used up all our glycogen. Then our body will start to do this. So, but we get so much ATP from it. Why is this not the main source of energy? Why is this not the main source of metabolism? I want you to think about that question. Oh, Maggie's going to actually just jump right on it. Maggie, what were you going to say?
I was gonna say, does it like not last longer, or
Which one? Which one lasts longer?
the fatty acids don't last?
When you say they don't last, what do you mean?
Like they run out, like the energy doesn't last as long.
Well,
Like it
we're
breaks down
making,
faster.
but we're making like triple the amount of ATP just from one fatty acid. With glucose, we only generate like between 33, 35, 38 ATP.
Does it have anything to do with like how like I took a sports nutrition class last month and we were talking about how like depending on what energy system you're using, like when you're working out,
Mhm.
like your body prefers one more than the other. So like if you're working out for if you're doing like more endurance type thing, something that lasts longer, it's going to rely more on like carb breakdown than fat breakdown. So does it have anything to do with that?
It probably does. Here's my immediate answer to this is a multifaceted layered thing. There's not one specific answer for why we use glycolysis as our immediate energy source instead of beta oxidation. There's many different reasons, okay? Yesterday I said this, but glucose is the brain's preferred energy fuel, like source of fuel. That is what our brain cells, okay? That is what they prefer. We have to have glucose for our brain cells. Think about this. The food we eat contains carbs. It contains glucose, right? Our diets are heavily carb diets. A lot of times, unless you're specifically trying to avoid carbs. But just inherently, a lot of vegetables contain carbs, okay? Not just breads, all kinds of food contains carbs. And so we have to have a way to metabolize carbs, right? If those are, that's a macronutrient that we're feeding our body a lot of. We have to have a way to metabolize it, okay? Think about this. Beta oxidation, there's a lot more steps to breaking down the triacylglycerols, to manipulating them, activating them, to transporting them. There's a lot more steps getting them out of storage than there is from the immediate digestion. The digestion of fats is a way more complicated process than the digestion of a carbohydrate. I mean, just look at the molecules. I'm drawing an example here. I mean, okay, palmitic acid versus, let's draw glucose. I probably drew these OHs wrong, but anyways, so a monosaccharide versus a fatty acid, which one's more polar?
The monosaccharide.
Yeah, so it's going to mix in our bloodstream better. It's going to be able to be quickly digested because it just mixes with water, simply because it's polar. Fats are majorly nonpolar. So again, that's why we walk through the digestion of fats. It's a lot more complex. There's more steps. We have to have all the transport molecules to help emulsify them and all the stuff. So it's just a more complicated process because the nature of the molecule than it is with molecules like glucose. Okay. So, but we get like triple the amount of energy. It's just really cool. So anyways, let's finish up these last two slides. So let me me scroll back up actually. Just from the breakdown of one stearic acid, we said we would get 9 acetyl-CoA. And we know acetyl-CoA, we come down here, acetyl-CoA feeds directly into the citric acid cycle. And then we run that and electron transport, right? Now, I'm gonna read off this slide for just a minute. The steps in the enzymes of beta oxidation run faster, I'm gonna highlight that, they're faster than those of the citric acid cycle. So what that means is if we are depending on beta oxidation, we're short on glucose, we've deprived our body of all the glycogen and glucose it has, so now we are metabolizing fats for energy. So if we're running this cycle, we're going to make a whole lot of acetyl-CoA, and then they're gonna move through the citric acid cycle slower. So we're gonna have a buildup of acetyl-CoA. There's gonna be a bottleneck here, okay? So if too much acetyl-CoA is made and a buildup occurs, then acetyl-CoA is converted in liver mitochondria to ketone bodies. And we did introduce this idea yesterday, but we didn't look at it in detail. But it happens through the pathway of something we call ketoacidosis or ketogenesis. So that's where the name of the ketogenic diets come from, is because our bodies are now resorting to this pathway. We get a lot of acetyl-CoA. We don't want too much acetyl-CoA. So we're going to convert it to ketone bodies. And this was the figure I showed you yesterday, an example of 3 different ketones. And these are dumped directly into our bloodstream. If you noticed, especially with these two, the 3-hydroxybutyrate and the acetoacetate, these have, they're carboxylic acids, right? They're not just ketones, they're carboxylic acids. And so they're acidic. So guess what they do in our bloodstream? They can make H+, which can react with these to make acetone, but they can make our bloodstream more acidic. Okay, so the danger with ketone bodies can ultimately lead to, I'm reading down here now, 2 of the ketone bodies are carboxylic acids. Thus, in prolonged starvation or in untreated diabetes, buildup of ketone bodies in the blood leads to something we call ketoacidosis. Okay, and this is where ketoacidosis, due to the buildup of ketone bodies in the bloodstream, the pH of your blood steadily drops lower and lower and lower, and that's fatal if it drops too low without being corrected.
It starts to shut your organs down.
Yeah. So why did we relate this to diabetes right here? It says, or in untreated diabetes. I think this is a homework question too. What happens in diabetes? Diabetes is an issue with insulin.
Like, why do diabetics go into ketoacidosis?
Yeah.
Oh, because, like, I mean, you have too much blood, like you have too much glucose in your blood and you don't have insulin to break it down. So it's just running through you, and your blood glucose is too high for too long, then I guess more ketone bodies are going to be made. And
so remember right here when we were talking about insulin, we said one of the roles of insulin is that it speeds up glycolysis, because we have, yes, like Robin said, a rising blood glucose concentration, too much glucose in your blood. In diabetics, if it goes untreated, we have massive amounts of glucose in the blood and we don't have insulin working. So we're not triggering the body to run glycolysis and break it down and get it out of the blood or do the other things that it does. Okay. So glucose is just staying in our blood and we're not running glycolysis. Okay. So if we're not running glycolysis, we're not getting ATP. We're not making ATP from glucose. That's what glycolysis does. So we're using fatty acids as our energy source. and we're metabolizing our fatty acids. This is why a lot of times untreated diabetics lose a ton of weight, even though they may be eating plenty of food. And that's where you get the buildup of acetyl-CoA and then the production of ketone bodies. Okay, so definitely know how diabetes is related to ketoacidosis. Definitely know that. I think that's a homework problem anyways. All right, last slide and then we're done. Biosynthesis of fatty acids. So earlier we talked about the storage of fatty acids and the metabolism or the mobilization of fatty acids. So we've looked at the whole mobilization part and the metabolism and beta oxidation. What if we don't need to metabolize them though? What if we have all these lipids in our body and we just need to store them? This is where we can take acetyl-CoA and we can make lipids, okay? And several different pathways feed down to acetyl-CoA. So ultimately, this is how, again, our biochemistry is all connected. We could take protein metabolites, amino acids, and run them to acetyl-CoA and make lipids. We could take glucose. Glucose could ultimately feed into the synthesis of lipids. And we call this lipogenesis. Remember the word genesis yesterday? I said means creation. So the creation of lipids, lipogenesis. This is the biochemical pathway for the synthesis of fatty acids from acetyl-CoA. So I want you to know that acetyl-CoA is our reactant starting material here. Okay, so as the end product of glycolysis and amino acid catabolism is acetyl-CoA, lipogenesis allows the body to divert instead of waste energy. Let's say we have too much protein. Let's say we have too much glucose. We don't just have to waste that stuff. We could just store it as fat. Okay, that's again, this is how people who continuously overfeed their bodies of too much glucose, too much protein, too much stuff. It just generates all the fats. Okay, this is what our bodies does with it, so that we don't waste anything. Okay, so anyways, I'm not going to run through these. This is the technical steps in the synthesis of this, but I really, guys, the reason I'm not doing it is because I want you to focus on beta oxidation. I want you to know glycolysis and I want you to know beta oxidation for the exam. Okay, so that is today's lecture. Questions?